When normal cells are introduced into a petri dish at low numbers, they begin to divide and proliferate. As the cells begin to touch one another, they slow their rate of division. This behavior is a consequence of the process called contact inhibition. Once the cells fill up the bottom of the dish, the rate of cell division slows further and is balanced by the rate of cell death, such that the total cell number remains constant. This state is called confluence. Contact inhibition ensures that the cells create a layer only one cell thick, a monolayer. The behavior of cancer cells is quite different. If a cancer cell is seated among normal cells, all of the cells will proliferate as before. However, once confluence is reached, the normal cells will regulate their growth, while the cancer cells continue to divide in an unregulated manner, yielding a clump of cells, which is often called a focus. Contact inhibition can be demonstrated in vitro by removing cells from a confluent monolayer. In this experiment, cells are removed by scratching the monolayer with a needle. The surviving cells at the edge of the wound now do two things. One, they begin to proliferate more rapidly, since they are no longer fully contact inhibited. And two, they migrate into the empty area of the wound, attempting to fill it up. As a normal part of growth and development, the body must generate new blood vessels to oxygenate the tissues. In a process called angiogenesis, new vessels sprout from existing ones. In this movie, we see endothelial cells sprouting to form new branches from the aorta of a zebrafish embryo. Each sprout is initially formed by one or a few endothelial cells. The process begins when an endothelial cell of a small vessel is activated by an angiogenic stimulus, such as vascular endothelial growth factor, or VEGF. In response to the stimulus, the endothelial cell becomes motile and extends filopodia that guide the development of a capillary sprout. The leading or tip cell continues to move away from the capillary as cells behind it migrate in and divide, forming a stalk. The sprout begins to hollow out, forming a tube. In this process, pinocytic vesicles fuse with one another. The large vacuoles formed in this way then fuse with one another, creating a lumen that runs through the capillary sprout. In culture, endothelial cells behave in a similar way. They spontaneously develop internal vacuoles that join up from cell to cell, creating a single lumen shared by many cells. In the example shown here, the individual cells contain either a red or a green fluorophore. Note that the areas of green and red are distinct. Even though cells share a lumen, they do not share cytoplasm and remain separate cells after the fusion events. Angiogenesis is critical not only in normal development and wound healing, but also in the development of tumors. A tumor must stimulate blood vessel formation to grow more than a few millimeters in size. VEGF is a key activator of angiogenesis in both normal cells and tumors. When cells within a tumor become oxygen deficient, they begin to express VEGF. VEGF diffuses through the tissues, activating endothelial cells on nearby vessels. This results in capillary sprouting. Some new cancer therapies are targeted to block the action of VEGF, with varying clinical results.